hopefully, recording in progress. Hopefully, people will start to filter in, either online or even better face to face. So we're all down here in the hub today. I think this is our first CCS seminar in the hub. You know, we've got plenty of empty seats for those who are wondering if there's a spare seat for them. So thanks, Jeff. So please, if you're sitting in your offices and Zooming this, please come down and join us face to face. Absolute stellar CCS seminar today. There will be lunch afterwards. So PhD and honours students, free lunch. It's a good reason to come. And you can't have it unless you come and attend the lecture face to face. So before we start, I'd like to acknowledge the Burrong and Wurrung clans of the Eastern Kulin Nation, who are the custodians of the lands and waters on which we're meeting. And I pay our respects to their elders past, present and emerging. So for this seminar, we have the Department of Anaesthesiology and Perioperative Medicine, who is really one of our absolute stellar world-leading research departments in the Central Clinical School. And the lineup's headed by Professor Paul Myles, who is undoubtedly one of the rock stars of Australian medical research. New England Journal of Medicine after New England Journal of Medicine paper for multi-centre pivotal clinical trials. So Paul, for those of you who don't know him, Paul, which would be hopefully not very many, is the Professor and Director of the Department of Anaesthesiology and Perioperative Medicine at Alfred Health and Monash University Central Clinical School. He's an NHMRC Leadership Fellow and a Fellow of the Australian Academy of Health and Medical Sciences, and also a member of the US National Academy. He's the editor of the British Journal of Anaesthesia and editorial consultant for Lancet, published more than 350 papers, most in the New England Journal of Medicine, and has been awarded more than 20 NHMRC grants worth more than $30 million. And his main focus of research is on patient quality and recovery after surgery and avoiding complications. So Paul is going to kick off the session by talking about reverse translation in perioperative clinical research. Paul.
Thanks, Gary. I enjoyed it, nevertheless. I want to take you back to 2006. The Cooksey report was released in the UK. It was effectively an economic review of the return on investment for medical research in the UK at that time. And there was a big sort of emphasis on these so-called gaps in translation, the so-called first gap, second gap, both in terms of bench to bedside translation of research into clinical practice, but equally into health policy and service delivery. There's this concept that science or medical science, medical research sort of has this kind of transitional process that goes from left to right. That may be the case for a lot of new discovery research and so on. But my interest is really about reverse translation. What is it can we learn from clinical practice or clinical trials and bring it back to the laboratory to actually investigate perhaps unexpected or interesting findings that we sometimes find. I want to base this talk in large part on all the experience at least I and members of the ANSCA Clinical Trials Network have had over now more than 20 years. We've enrolled, in fact, even in these studies here on this slide, more than 30,000 patients into multi-centre international randomised trials in surgery perioperative medicine. I want to focus on just one of those trials, what we call the Atticus trial, aspirin tranexamic acid for coronary artery surgery, and in particular, the effects or benefits of tranexamic acid in cardiac surgery. When we set this study up across Australia and around the world, this drug was being used in roughly 50% of all open heart surgery cases, and that use varied widely from surgeon to surgeon, hospital to hospital, and different parts of the world. And we really wanted to define whether or not there was a net benefit with this drug. So it was a really successful trial. It took a long time because of the complexity of cardiac surgery and the changes in practice that happened across the 2 NHMRC grants I got for that study. But we enrolled over 4,500 patients having cardiac surgery across 31 hospitals in 7 countries. And these are the main effectiveness or efficacy findings from the study, we clearly demonstrated that this antifibrotic drug, tranexamic acid, reduced the amount of blood loss during and after surgery, the need for a blood transfusion, the number of units of red cells transfused during and up to a week after surgery. And perhaps most importantly, it halved the requirement for emergency reoperation for surgery, most often because of cardiac tamponade or excessive hemorrhage happening in the ICU ward in the hours and days after the operation. I can tell you, for somebody who's done more than 35 years of medical on-call here at the Alfred and elsewhere, there's nothing worse than at 2 in the morning getting rung up to say a patient needs to be rushed back to theatre. It has high costs, it's incredibly socially disruptive for all the staff involved, and it adds great risk to the patient's eventual outcome in that setting. So these findings were really important. And I just want to sort of remind you now of a simplistic sort of sense of what the biology of tranexamic acid is. It's labelled an antifibrotic drug. It's a lysine analogue, and it actually binds to the lysine receptor on the plasminogen molecule, stopping it from binding to fibrin, leading to further activation of plasminogen into active plasmin. That's simply how it works. And that then brings me to this part of the story. The wonderful Rob Metcalf from ACBD here at the Central Clinical School. He listened to some talk I gave on tranexamic acid many, many years ago, and came up to me after the meeting and asked me, you know, you sort of described the actions of tranexamic acid and talked about it as an antifibrotic drug. Do you realise that it's not just fibrin-centric? That the underlying purpose of plasmin, or the plasminogen biological system, is partly to actually deal, it's a protease of course, not just a blood clot, but also dead proteins that are being generated all the time in the body, that in fact there was pro-inflammatory activity of plasmin and is likely to affect the immune response. Now, I didn't know any of this. I am an anaesthesiologist, clinician, and worry about bleeding, but of course these other effects. And in fact, you know, most of you will know that Rob Metcalf's done a lot of work in his lab looking at the whole biology and potential effects of plasmin across different areas of medicine and disease states. We know that in fact it's important, or plays an important role in complement activation, in the induction of cytokines, cell signaling, and so on. And in fact, there's a lot of studies that come out from lots of groups around the world that really sort of suggest that overall it's likely that clearly, because of this pro-inflammatory effect of plasmin, which is activated in major surgery and trauma, that it's likely to have a net immunosuppressive effect. So Rob said to me, look, are you looking at infections in the ADEX trial? Because our whole interest was bleeding and bleeding complications and thrombotic or pro-thrombotic risk. And of course we hadn't. But given all this work that he had done and the science that he understood, we then immediately started collecting infection data for all patients in the ADEX trial that were being enrolled at that period of time, right up until the end of the trial. Well, the other effect that he was interested in investigating was, well, given that plasma is likely to be pro-inflammatory, perhaps immune suppressive, what's the effect in healthy volunteers? And in fact, no one really knew this story. So we had to go through ethics committee and put an ad up around the hospital, and 10, mostly partly medical students and junior doctors, volunteered to have a 1 gram dose of tranexamic acid. They had baseline bloods taken off first, and then we repeated the blood sampling at 2, 4, and 24 hours after surgery. So these are totally healthy people, young, healthy people going about their daily jobs, looking at these cell markers and activated processes. In actual fact, we found very clearly that, in fact, even in this completely healthy young population, that the effect of tranexamic acid was that it did, in fact, reduce pro-inflammatory cytokines, immunosuppressive cytokines, immune activating markers, and so on. So the bottom line of that is, at least in this preliminary evidence, that tranexamic acid is likely to be immune enhancing, and that would be really important in major surgery. So we then came back to the question, well, does it in fact reduce infections in open heart surgery? So we therefore nested in this planned sub-study and collecting all infection data of patients who were being enrolled in the study from that point forward. And we looked at using the agreed census of diseases and control criteria for healthcare associated infections and surgical site infection. And these are the key results. We had over 600 patients in this nested sub-study of the main trial. And as you can see here, there's, I guess, a hint, a trend, that in fact there could be a reduction in infections. This is a randomized trial, double-blind randomized trial in cardiac surgery. So that got us at least interested to pursue this story further. And the other thing that Rob said to me, which is again quite a few years later when we were actually starting to look at the main results of that infection sub-study, and he said, did you collect any information about diabetes status in the trial? Are these patients having cardiac surgery? So of course we collect their comorbidities, and we knew whether or not they were diabetic or not. And the key thing about this is he understood, which again I didn't, was that plasminogen is glycated in diabetes, and the more poorly controlled the diabetes is, the greater the extent of that glycation. It's the underlying reason why HbA1c is a marker for diabetes control. And he reasoned that, or maybe tranexamic acid won't work as well in diabetics. So that is just obviously a hypothesis generating question that came from the science. So we then looked specifically at the non-diabetics to see what their infection risk reduction was. And as you can see here, that in the non-diabetics, where there is no glycation of plasminogen, that in actual fact, the effect sizes were stronger, and some of these were, of course, statistically significant. In fact, all the point estimates were really supporting this reduction in infection risk. So we then said, okay, what about if we go back to the full AGAS trial with the full results and actually look whether or not those bleeding complication reductions that happened across the trial was stronger in the non-diabetic population. So this is the full data set. We broke it down into non-diabetics and diabetics. And as you can see here, the non-diabetics again had a stronger effect of the tranexamic acid. So we're starting to get a bit of a story now that's linking together from, obviously Rob Metcalf's superb understanding of the biology of plasmin and so on, and this potential effect modification of diabetes. And we've actually published all of those findings, including all the basic science that went into it in Blood Advances back in 2019, where we clearly demonstrate quite a lot of, biological plausibility for pro-inflammation and immune suppression effects of plasmin itself, and how tranexamic acid may well suppress or improve effectively the status of the patient having major surgery. And the second bit is actually even more important for us in perioperative medicine, because we already know that diabetes status is a risk factor for infections and other complications after surgery. And it's well accepted practice that we will always try and first of all diagnose and then better treat or stabilise diabetes for patients that are coming in for elective surgery. But we've now got a completely different and equally important reason why that is so crucially important. Because if in fact poor diabetes control adds to the, either pro-inflammatory effects of activation of plasminogen, which happens in surgery, or in fact in circumstances where you're using tranexamic acid to block that effect, it won't work so well in poorly controlled diabetes. So people having hip and knee replacement surgery or trauma surgery, where it's crucially important to avoid infections, this would be really important. So therefore, we're investigating these concepts further in a specifically designed large randomized trial, because of course this would be clinically important if proven to be so. So we convinced the HMRC again that they should fund this large randomised controlled trials. We're enrolling over 3,000 patients having major GI surgery, again as a multinational study around the world. The infectious diseases expertise is coming from one from Trish Peel. Rob Metcalf is doing further lab work with a lot of specimens that we're providing. And of course, we've got the involvement of Wendy Brown, who's Professor of Surgery here and equally a leading upper GI surgeon across Australia. And that's underway at the moment. So 3,300 patients are being enrolled in the study. We've got specific aims that we're aiming to investigate, and we do hope to further progress this story. Now, I just want to move you sideways a little bit because Rob Metcalf actually knows quite a bit about quite a few things related to plasma. I didn't know that plasma is the key mechanism in blood-brain barrier breakdown that happens in some disease states. And his research, mostly his interest is around traumatic brain injury. And that sort of disruption and leakage of the brain with cerebral edema and so on, which is a severe clinical problem, seems to, at least in part, be activated or worsened by plasma activation. So that's a story that's, certainly he's researching quite a lot of at the moment. I also now want to introduce Rob Sanders, has published a paper in this month's issue of the British Journal of Anaesthesia. Rob is a neuroscientist. He did his honours in neuroscience at Imperial College, then went and did medicine, then became a specialist anaesthetist, then got headhunted to the UK. I tried to headhunt him here, but Monash didn't have any money at the time for it.
You're still not, Paul.
What a perfect fit for the Central Clinical School. So sadly, he's been appointed professor in Sydney, but I have not stopped my interest. And in this month, he's got a big interest around cognitive impairment, particularly delirium and ongoing cognitive decline after major surgery. And in this study, one of the things he looked at was patients who were either had post-operative delirium or didn't after major surgery. Took blood samples and CSF and looked at the CSF blood ratio of albumin, which is a marker or a measure of breakdown of the blood-brain barrier, clearly demonstrating that in post-operative delirium, there's an intact blood-brain barrier, but that's clearly broken down in patients who are delirious. And that is, again, a very big problem in hospital medicine. So the question then becomes, well, if that seems to at least in part be generated through plasminogen, can tranexamic acid block or protect the blood-brain barrier to reduce the risk of delirium after major surgery? So we've now set up a nested sub-study of our NHMRC funded trial. We're expecting to get funding from the college literally in days, I hope. And in this, we're specifically doing all these cognitive measurements and otherwise done before and after surgery, out to a year after surgery, looking at both the incidence and severity of post-operative delirium in patients having this major GI surgery. Now, I've been talking about an antifibrolytic drug, or if you like, plasmin and fibrolytic pathways. And of course, the first use of a fibrolytic drug in clinical medicine was streptokinase in the late 1960s. It's now a standard of care or its derivatives to treat myocardial infarction, stroke, peripheral embolism, and so on. What I didn't know, or hadn't sort of thought through as a medical student, was that streptokinase of course, is generated from a streptococcus that in fact was first reported way back in 1933 as being the mechanism or a mechanism of how the beta hemolytic strep actually break down the clot around a local trauma or infection source so that the bacteria can spread through the tissues. It's very sneaky. It's a great survival and dissemination mechanism. And here at the Alfred Hospital, we are the referral centre, statewide referral centre for hyperbaric medicine and all cases of necrotizing fasciitis, which is a group A strep infection. It's a devastating, ugly, uncontrolled infection that just eats away and spreads through the tissues in a very accelerated rate, needing massive surgical debridements, a long stay in ICU, repeated hyperbaric oxygen treatments, and does have, of course, a very poor long-term disability-free survival. And the mechanisms by which necrotizing fasciitis first begins and how it spreads through the tissues is through these proteases, which very much includes activation of plasmin, leading to the more severe forms, such as in those pictures. Now, in actual fact, the role of fibrinolysis in these strep infections is probably far more powerful than many people had really considered. This interesting paper came out in 2014. Again, I was alerted this through Rob Metcalf. And in it, really, they talk about the fact that the strep infection actually sequesters the host plasminogen, leading to its secretion of streptokinase and probably other proteases, so that it can actually migrate through the tissues and cause a much more extensive and formidable infection. And, of course, tranexamic acid blocks plasma. Could it be that tranexamic acid, an incredibly cheap, incredibly safe drug, can actually be instituted early in this terrible condition? And in fact, again, there's some biological plausibility for this. This paper came out in Scientific Reports in 2017, where they're looking at some fatty acids, but also tranexamic acid, to see whether or not they can suppress these invasive strep infections. So, in fact, right now we've got a feasibility trial that we've had approval through ethics. We're linking in with a hospital, the hyperbaric unit in Sydney, and also Darwin Hospital, which the Indigenous community up in the Northern Territory have a really high rate of necrotizing fasciitis, for which treatments are literally, minimal in that setting. They don't have a proper hyperbaric service, for example. And we just want to see whether or not we can do the trial, enrol patients who are critically ill, and ideally at an early enough point in their disease to actually turn the corner in the spread of infection. So that's only honestly just begun. Now, I went through the whole talk and not once talked about COVID, but I can't help myself because we're actually currently communicating with a US venture capital company, a spin-off of University of Texas, where they're interested in looking at effectively a nasal spray of tranexamic acid based on the work that Rob and I published in the last couple of years in this COVID pandemic. Because there's good rationale of why blocking plasma may in fact be a potential treatment for COVID, and certainly the escalation of the disease in its early phases. Plasmin is crucial to the cleavage of the spike protein for the coronavirus, for it to be able to bind to the ACE receptor and so on and get into cells. I've already told you it blocks the pro-inflammatory and immune suppressive effects of activated plasma. And we also know that, in fact, the 2.5% concentration, this is from other people's work, which is a quarter of the concentration we're currently using in clinical practice right now, actually suppresses other pathways that may be tied in with plasma. So that's my little sort of spin around my interest in reverse translation. I think some of the clinical studies that ourselves are doing, and many others, I guess, across the central clinical soil, I think there's a lot more opportunity for us to actually engage more closely with the different labs that are here, and perhaps even collaborate on some of the concepts we're already exploring at the moment. Thanks very much.
Good bit of time for a couple of quick questions. Now, people online, I can't really see who you are, so I think just turn your video on and ask a question if you've got any. Do we have any questions in the room?
Thanks, Paul. It's absolutely fascinating to hear all that. And as you know, my interest is in translation in general. I'm wondering if you had any comments about how reverse translation could be enhanced. Like, would there be anything, mechanisms we could put in place to ensure that happens more effectively? Or have you got any thoughts?
Absolutely. I think, on our TRIGS trial, we have consumer representatives on the study, for instance, and they are giving us advice around the sort of things we're measuring, the sort of things we're worried about, and more importantly, they're telling us what, as patients, what they were worried about having major surgery. And they certainly tell us or reaffirm the importance of avoiding infections and so on. But I think that can apply a bit more broadly, because if we publish some clinical study of any sort, a trial or whatever, and we all think of all our clever ideas and scientists will think of theirs, I think we're in bubbles. And I just think the consumers, healthcare policy makers and others will be wanting to know different questions like, well, how does this, first of all, how do we know it works this way, and what other things might happen? What will be the side effects? You've told us about the positive benefits, could there be side effects? So at that level, but more importantly, when we come to show that, maybe demonstrate there is a benefit in reducing infections, how do we spread that word more broadly across the healthcare system, into policymakers, guidelines, and so on? So I think there's a lot of role for that. And again, we just keep thinking it's one-way process, and it needs to be a lot more continuous.
Sorry, can I just follow up? So it seems like also what was really crucial was the discussion with the other research group. So maybe it's also about enhancing that kind of cross-disciplinary.
Yeah, exactly. So I gave this talk here today for this very reason, because the whole purpose of the Central Clinical School Symposiums are to tell others about the research you're doing, and for others to think, well, how can I perhaps use them or work with them, or that they help us in that two-way process. In fact, this whole hub development, including the little social cafe, is specifically designed for this exact reason, for us to actually tell other groups what we're doing. And in my case, it's actually, I mean, I'm aware to a degree of some of the labs and so on. I just think there's so much missed opportunity at the moment.
One of the reasons why we've decided to have these in the hub, so hopefully in future we'll get more people. Just one, is there any other questions either online or? Yeah.
I guess when we're thinking about getting funding applications, there's a very traditional approach through NHMRC where you do the basic science, then you do the early testing and things like that, and then you put it out into a large clinical trial. Do you have any sorts of thoughts on how we can target when we're doing these reverse engineering? How can we, because we don't have necessarily, we do have some of the basic science, but not the complete handle. We're exploring it as we go. Is there some advice or some way you go about trying to convince the funders? Obviously, you've been quite successful. How can you, it's not your traditional way of approaching funding, so how do you bring the funding bodies on board?
Well, I think the most important thing is to demonstrate the value of this type of approach more widely and tell the stories where, in fact, there is value added to an NHMRC grant for a trial, that there's spin-off benefits. In other words, a research agenda is expanded and developed in ways that perhaps would never have happened. Now, some of these are going to end up dead ends. I mean, I'd be astonished if we're going to cure COVID with tranexamic acid, but it's worth pursuing it because, again, that's a cheap, simple, safe drug. You know, we have generally not been successful at NHMRC and certainly MRFF by going back and wanting to do these sort of follow-up mechanistic studies. They're expecting effectively us, like always, to do this for nothing, come back to them with a clinical trial or something. But that's not to say that we shouldn't keep trying. I think the more stories, the better. Certainly at the next level down grants, and in our case, the College of Anaesthetists are very generous. They give out $1.5 million of funding every year. We can certainly go to them, and they are very keen to support this type of is translation, but in our case, cycle research. Yeah, because they like this idea of discovery and building in clinician researchers with basic scientists. But I just want to keep going. I mean, you know me, I do not give up. Do not give up. Just keep putting grants in.
Yeah, and I can see the success of that strategy. If you're going to survive in this business, you've certainly got to be determined, don't you? Yeah. And resilient. And it is, I understand that the NHMRC leadership grants are going to be released today, maybe even while we talk, which might be why people are not in the room, because they're huddled around their computers hoping no one talks to them until they find the results. Thanks, Paul, very much for that. Next we move to the earlier end of the career stage, and another rising star, Dr. Jennifer Reilly, who's a specialist anaesthetist at the Alfred and the Central Clinical School, whose research includes risk prediction, registries, and big data. Her PhD thesis was titled Predicting Perioperative Mortality in Adults Undergoing Non-Cardiac Surgery in Australia, and she's developed a rich calculator to predict mortality amongst adults having non-cardiac surgery based on the surgical outcome risk tool in the UK and its New Zealand adaptation. So Jennifer's going to talk to us about risk prediction and data-driven perioperative care. Thanks, Jennifer.
Thanks very much, everyone. I'm going to present today a talk that I was invited to give a couple of weeks ago at the Victorian Perioperative Clinical Council. That is the state advisory council to the Victorian Health Departments and consists of senior surgeons and anaesthetists across Victoria. Wendy Brown is head of the surgical subcommittee of the Victorian Perioperative Clinical Council, and this was their annual workshop that was presented for the council as well as for the health department and Safe Care Victoria and clinical leaders across Victoria. So the talk was on risk prediction and data-driven care, and I'm going to just present to you today the first two parts of that talk. So an introduction to risk calculators and about my work developing an Australian perioperative mortality risk calculator. I do want to just go into a little bit of the clinical context though, so that you can understand how this work might apply in Victoria. So coming out slowly of the pandemic, baseline surgical volume in Victoria is currently 160,000 cases, or it was before the pandemic. And surgery volume is going to increase from baseline by 25% or 40,000 cases in 2023, and then by 50% or 80,000 cases a year in 2024. And then it's going to be held stable at 240,000 cases per year after that. So we see a significant increase coming up. This plan is going to be delivered in two main ways, through the public and the private sector. In the public sector, we're going to see more same-day surgeries, not just an increased number of the usual surgical procedures that we perform, but we're going to see a shift to perform some inpatient procedures moving to be performed as same-day procedures. So where people would have usually stayed in hospital at least one night, we'll be looking to send them home on the same day. It's going to be achieved by performing more nighttime and after-hours surgery, and by increased efficiencies and fast-tracking patients through the health system. In the private sector, we're going to be doing an extra 50,000 surgeries, sending public patients across to the private sector by June next year via these public and private arrangements. And a total of $1.5 billion is being spent on this program. So this includes about 500 million in the public system and about 500 million in the private system, and another 500 million on the supportive measures such as staffing and equipment. Sorry, I think Paul's slides were still showing for those on Zoom, so mine are now up. Sorry about Sorry for those who missed the first slides. So with an extra 80,000 surgeries per year in Victoria, if we assume that half of these are going to be inpatient surgeries, then we need to anticipate an extra 8,000 major complications in Victoria every year. This is based on a simple model where we know that 20% of there is a 20% major complication rate in adult inpatient non-cardiac surgery, both in Australia from the Reason study done by ANZCA investigators, and also internationally from the International Surgical Outcomes Study group. So this is 8,000 extra major complications per year from 2024, which is 31 extra major complications on every weekday, or 22 every day of the year. So thinking about the implications of these, we need to start identifying our high-risk patients in advance. These patients are going to be unsuitable for the public and private lists for the surgery that is currently done as inpatient surgery, and is now planned to be outpatient surgery, so going home on the same day. They're going to be unsuitable for hospitals that don't have 24-hour medical coverage, that don't have on-site diagnostic facilities or physician input, and that don't have on-site critical care facilities. We need to think for these patients. We know that high-risk patients have an extended PACU stay, or the recovery unit stay. They have a longer hospital length of stay. They have increased critical care admissions, increased medical emergency calls, and we know that there are increased medical and legal claims in our highest risk surgical patients. We need to be thinking about 8,000 potential episodes, extra episodes of bed block every year, because we know that these patients have longer length of hospital stay. So we're going to have bed block while we're trying to increase our surgical admissions by 50%. We know that in our over 70s, the 30-day mortality is 5% here in Australia. What we don't know is what our 3, 6 and 12-month mortality is. We need to think about, are we adding value in these patients? And there's a huge opportunity for a cohort study across the health system. We also need to consider the impact of unplanned return to theatre and readmissions. So that's the clinical context for the talk, with the slides that I'll talk to you now about a background to risk calculators and then development of a calculator. So the Knowing the Risk study was based on a snapshot of every surgical patient in the UK over 1 week in 2011. The Knowing the Risk study showed that high-risk patients make up 20% of the UK surgical population, but 80% of perioperative mortality. High-risk patients make up a large amount of the surgical workload on the wards, and they consume an oversized share of hospital resources. Half of the high-risk population in this study were elective surgery patients. This report identified 4 broad steps to address risk in surgical patients, because it found that 1 in 5 high-risk patients in this study went to critical care, and 50% of high-risk patients who died did so without accessing critical care resources. One of the key recommendations was a need to introduce a system that allows the rapid identification of patients at high risk of morbidity and mortality. The report states that while this might seem obvious, the literature is full of differing descriptions, scoring systems and tests to meet this aim. Another one of the recommendations was that an assessment of mortality risk should be made explicit to the patient and recorded clearly on the consent form and in the medical record. A third recommendation was that the decision to operate on high-risk patients, especially non-elective patients, should be made at consultant level and should involve surgeons and those who will provide the intra and post-operative care. So this suggests intensivists should be involved in these decisions as well. And a final recommendation was that once a decision to operate has been taken, that we need to provide a full package of supportive care. And crucially, that if critical care admission is recommended but is not possible, then this should be disclosed to the patient as part of the informed consent process. The surgical outcome risk tool was developed using data collected by anaesthetists during a 1-week national surgical outcome study. The sort has high predictive performance, and it's available as an online calculator and a free smartphone app that's shown here on the screen. The advantages of this calculator are that it has only 6 variables, and they're all simple clinical variables available preoperatively, and it doesn't require any laboratory tests. Now, this calculator was actually developed using the data from the Knowing the Risk study, and this is the tool that I've set out to adapt for Australia. My colleague Doug Campbell in New Zealand undertook a study of SORT in over 300,000 patients in New Zealand using their national minimum data set that's collected on every surgical patient. He found that SORT underpredicted mortality up to 5-fold in some risk groups, and this really highlighted that risk prediction models cannot be exported from one country to another without being adapted or updated using a data set from the new country or population. Doug updated SORT for the New Zealand population, and his calculator was released as NZ risk. He's added 2 extra variables into this calculator. So these are sex and Maori or Polynesian status. And he found that the risk prediction was accurate not just at 30 days, but all the way out to 1 and 2 years. PPOSM is a risk calculator that many surgeons and anaesthetists will be familiar with. There is one published Australian study that attempted to externally validate this calculator in an intensive care population. The authors found that the original model significantly overpredicted the risk of death and suggested the model would need to be recalibrated before it was used routinely in Australia. From an anaesthetist perspective, the PPOSM requires 18 different variables, including 5 laboratory variables that are often not yet available when we see patients in the pre-op clinic. There are also at least 4 post-operative variables in the, 4 operative variables in the model. So if we're using it pre-operatively, we need to estimate what they might be. It's limited to use in general surgery, but there are some specific models for other types of surgery. This is the NS-CRIP risk calculator, again, which surgeons and anaesthetists will be familiar with. The American College of Surgeons have published good data on the performance of this model, and the tool predicts not only mortality, but also major complications, and as well as admission to a nursing home after surgery. Another advantage of this tool is that it provides an indication of relative risk, which is really what we need to know. Where does our patient's risk sit in comparison with other patients? This calculator does talk about average risk, but really there is no average risk because when we look at how risk is distributed across the population, it's not evenly spread. We have a much smaller number of patients at the high end of the risk curve, but where we choose to cut the risk curve and to label our groups as low, medium or high risk is unique to each published study depending on its results and health system. In the NZ risk data, they decided to cut the curves in a way such that 80% of patients are called low risk, 10% are intermediate and 10% are high risk. And I think this shows that where we're heading with risk science is considering patients in deciles of risk rather than comparing to the entire surgical population. The NS-CRIP risk calculator that I just showed you that provides such lovely information is actually unfortunately a black box because we don't know the algorithm that's inside it. And this means that we can't replicate it. We need to regenerate it from scratch. The American College of Surgeons has been called upon to make the risk calculator available for external validation, but they've responded that the algorithm is proprietary and there are no plans to release it. Many clinicians will know that New South Wales public hospitals have recently joined the NS-CRIP program, but at this stage there are no plans for New South Wales to update the risk models and to make them specific to the New South Wales data. And if they did, they'd only be specific to public hospital data because the private sector is not included in the state program. So just to recap, SORT would need updating for the Australian population in order to be used here. PPOSM isn't calibrated here and doesn't apply to a large part of the surgical population. And we can't locally update the NS-CRIP here because the algorithm is unpublished. And even if we had it, we don't have a representative data set. We only have public hospital data from one state. But if we did have an Australian risk calculator, what might the patient experience be like? Well, we know from this study that patients like it when we use the NS-CRIP risk calculator in pre-op consultations to help their decision-making process and the consent process. This paper presents a survey of 150 patients on their experience of using the NS-CRIP risk calculator. It showed that 90% of patients wanted the calculator to be used again before any future surgical consent. High-risk patients were 3 times more likely to underestimate their risk of any complication, of serious complication, and their length of stay compared to low-risk patients. 70% of patients said that they would consider participating in a prehabilitation program to reduce their risk, and 40% said that they would delay surgery to do so. And use of this calculator also decreased anxiety in 71% of patients. If we had a risk calculator validated for the Australian population, it has multiple potential uses. Firstly, we can use it routinely to identify high-risk patients before surgery. We can use it as part of the informed consent process for surgery. We can then take it further. We can develop specific care pathways and clinical standards of care for high-risk surgical patients. We could use this tool to develop a national registry of high-risk surgical patients to generate evidence on risk factors, clinical outcomes, and patient-centred outcomes. And finally, we could use it to facilitate registry randomised control trials that are aiming to improve outcomes among high-risk surgical patients. So I'll just run through now my work on developing an Australian risk calculator. The literature review for my PhD was a systematic review of risk models that predict mortality in adults having inpatient non-cardiac surgery. We screened over 2,000 papers and came up with a shortlist of 21 models for consideration for adapting to the Australian population. And I then ran these models through a series of filters that are outlined here. I wanted the model to be useful for both emergency and elective surgery, and for all variables to be available preoperatively. I needed the model to have been studied in at least one other population in what's called an external validation study, as evidence that it was valid outside the model development population in at least one other setting. I wanted a model that could be reproduced using normal statistics rather than machine learning, because at that stage I didn't have access to the sorts of extremely large data sets that machine learning required. And this left us with 5 risk tools for consideration. Now, I mentioned before that I also needed the model to have a published equation so that we could study it here, and that NSCRIP hasn't been published. And so this tool was excluded and left us with 4 models. These models included SORT and NZ risk. There's a third model, the surgical mortality probability model, that contains variables that would already be collected during a study of SORT. So this could be studied using an existing data set collected for SORT. The fourth model, called the POS-POM, has a similar accuracy to SORT, but it requires 17 variables instead of just 6. And so this is why I've selected SORT as the basis of an Australian model. So a quick overview of my study. Our primary aim is to externally validate and locally update SORT, and to provide propose a final risk model for use here in Australia, and to examine its accuracy out to 1 and 2 years. A secondary aim is to test some new candidate variables, including socioeconomic status, which will be indicated by several Australian Bureau of Statistics indices compiled from national census data. I also want to look at rural, regional and remote geographic residence location, and potentially the Indigenous status. I had also planned to test frailty as a variable in the model, but for reasons that will become evident, including the pandemic, that will need to wait until a later version of the study. This study has been funded by grants both from ANZCA and the Australian Society of Anaesthetists, and the study has been formally endorsed by the Australian Commission on Safety and Quality in Healthcare and multiple specialist colleges, including the College of Intensive Care and the Australian College of Emergency Medicine, and it's also supported by the College of Surgeons. Initially, I started off with a pilot study. I wanted to establish the feasibility of collecting 1 page of baseline data at multiple hospitals across Australia. This was not going to be new data, but this was simply anaesthetists transposing clinical data that had already been collected onto 1 page of paper, single-sided, and submitting that to our study. So this would be data that collected all the risk variables, and we would then match this to death data. We got ethics approval to conduct this study with a waiver of consent. And we wanted to get feedback on the time it took for data collection, feedback on the structure of the form, and on the ability of the sites to readily provide the 30-day death data. Although we gained ethics approval to conduct this study with a waiver of consent, we found significant hurdles getting it through the site governance process at our 22 hospitals. To give you an example of these difficulties, one hospital took, I think it was over 250 or 270 days to give approval for us to collect this study. And at that point, they didn't request any changes to the protocol. And at one private hospital, the CEO refused to give us permission to undertake this study, despite approval from their own ethics committee. So we concluded that this project was not feasible as a research project in Australia due to the ethics and governance hurdles involved. So I started to consider alternative ways of collecting this data, and came up with the idea of collecting it within a registry. And so I established a pilot national perioperative outcomes registry called ANZCA PCOR Pilot. We were able to obtain a registry template from Susanna Ahern in the School of Registry Sciences, put together a modified protocol of over 100 pages, and submitted that to ethics for approval. We gained ethics approval here from the Alfred HREC for our pilot national perioperative outcomes registry. And then Site governance was rapidly improved at about 20 different hospitals with no particular hurdles or difficulties. And so we set up being ready to conduct our primary data collection through this new pilot perioperative outcomes registry at our 20 hospitals. The plan was to collect data on about 22,500 adults having inpatient non-cardiac surgery, and this would be conducted as a sprint audit over 1 month, similar to the anaesthetic sprint audits that they hold each year in the UK. We would collect the baseline sort variables in the anaesthetic bay by anaesthetists. The sites would provide 30-day mortality data, and we would match this to the National Death Index. And we were just planning to get started on this study when the pandemic occurred in early 2020. And so I embarked on an adapted methodology, which was to obtain a large national hospital administrative data set from the Australian Institute of Health and Welfare in Canberra. There was a reason that I hadn't considered this method, or I had considered this method previously and had chosen not to do it, because one of the key variables we need is the ASA score, which is the Australian Society of Anaesthetists classification score for patients. And if you ask if you ask AIHW whether they collect that score, they say no. And if you ask the Victorian Health Department and VAHI if they collect that score, they say no as well. And if you look at all of their data dictionaries, the score isn't mentioned and they don't collect the score. But fortunately, one of my PhD supervisors was Professor Belinda Gabay in the School of Public Health, who runs the Trauma Outcomes Registry for Victoria. And she was able to tell me that ASA is actually buried as, I think it's the 7th digit in the surgical code in the data. So in fact, that it is there and it was there all along. It turns out that it is missing in about 30% of patients, but we have developed imputation models to recreate that in some work that we've been doing alongside this process. And so I was able to actually return to what turns out to be a far better method using a large national administrative data set. This took an extensive process of negotiation with AIHW in terms of agreeing on the variables, going back and forth, justifying all of the variables that we wanted. 2 rounds of ethics committee approval, including the Alfred H. Rec, and then it had to go through the AIHW's own ethics committee as well. Ethics was approved in late 2020, and it took over another year to actually get the data delivered. I ended up handing in my PhD without this main study, which was meant to be the purpose of the PhD. So I conducted an external validation study using a different data set instead. And 1 month after my PhD was handed in, we did receive the data set, which was early this year. So we'll be embarking on the analysis in the next couple of months. In fact, I'm hoping to have that done by Christmas. Just to mention a couple of other projects that I'm working on regarding this data as well. This year I have 2 Masters of IT students through Monash University who are doing a project with us who are going to use I did collect some pilot data through the registry from sites, a 1-year data extract from about 6 hospitals. So we'll have about 50 to 100,000 patients in that data set, and the IT students are going to be developing. One student's going to be developing dashboards that would show us what a prototype registry would look like, and the other one's going to be developing a universal script that could be deployed at hospitals across Australia to allow them to easily extract a data set to pull into our registry. We're also going to be analysing and writing up our pilot data as an example of what could be collected if we did have a national perioperative outcomes registry here in Australia. We'll be hoping to present the risk calculator at least initially as an online web calculator, and hopefully down the track as a smartphone application as well. And then there'll be a whole body of work to do around replicating the patient survey I showed you to find out whether patients feel that the calculator is useful and how it can impact their care decisions, and then using it in clinical decision support systems to test out different models of care and different systems to see whether use of this tool can actually change downstream clinical outcomes. I'll stop there. Thank you.
Wonderful. Again, questions in the room, or if anyone online has questions for Jennifer, just turn your video on so you come up to the top of the screen and you see it, and yell out. Any questions? Right.
Yeah, I was interested in like you said, that the English British model was not applicable in New Zealand until they incorporated gender and racial background. I'm just wondering at what level of diversity becomes a problem. So would you, for example, need variations within parts of Melbourne or in parts of Australia to accommodate or to allow for the differences in populations in different parts of the country?
Thank you for the question. There's perhaps a subtle point that I skipped over. So first of all, they did a straight external validation, so they didn't change any of the variables, and they found that it wasn't accurate without including sex or the racial data. But in their model, they felt that it was important to include those things. They could have just changed the coefficients in the equation and updated the model, but they wanted to include those variables as well. They didn't actually publish the accuracy of their straightforward external. Sorry, they did actually publish the accuracy. I'm not sure that they got significant extra accuracy by adding those things, but they felt it was important to include those demographics in the model. As to whether we need things within a city, I think that would be too detailed, because if we did have a model for that, then if we had a model specific for Melbourne using certain suburbs, then you can't deploy that model in Perth, for example, or in Brisbane. So what we're looking for is a national model that we can use to implement in all of the states, and to do then national studies and even international studies with our colleagues who have locally adapted versions of the same model.
Can you just tell me a bit more about the concern by some Indigenous health people about the fact that there's a sense of, I think, concern that some Indigenous patients have been denied surgery because of the perceived excessive risk, where in fact an Australian-based risk calculator that has that data built into it could reassure and provide greater transparency.
Yes, sure. So I do have ethics approval to study Indigenous, to include Indigenous status in my model. That's in the main ethics approval, both from the Alfred and from the Australian Institute of Health and Welfare. We applied for a separate data set from the Northern Territory. So the main data set we can get from Canberra includes 6 states and territories. It doesn't include Northern Territory, and it doesn't include WA. WA have their own rules, and at this stage we won't be able to get data from them. But I put in a separate application to get essentially the same data set, a parallel data set from the Northern Territory. But they've had a change in the head of their ethics committee lately, and they're wanting to make sure that they comply with all guidelines around Indigenous research, and have advised that that means we need to undertake Indigenous consultation before we can include Indigenous status as part of our model and as part of our analysis. They recommended that I get in touch with the Indigenous Data Network at the University of Melbourne, which is an Indigenous-led big data research group focusing on health data. And one of the concerns in Darwin is that I think the head of the ethics, the new head of the ethics committee there is an intensivist, and has commented off the record, or at least not directly to me, that they are already seeing patients being denied admission to the intensive care in Darwin on the basis of predicted risk. So there's a concern that race is being seen as a non-modifiable risk factor, and that when we're including it, and that it's being used to predetermine outcome, if you like, and that they think it's already being used in their centre to deny people care. And there is a concern by them that we may then inadvertently end up denying surgery to Indigenous patients because of their risk, which is based, which includes race, if we build that into this calculator. Had a wonderful conversation with a researcher in Darwin who's done some work in this area on Indigenous risk in another field, and I think he said to me that the problem with this is there's no variable for racism and for systemic and structural disadvantage. So that part of the project I think is on hold, and Paul and I are going to catch up after this, but I'm keen to get on with an Australian model. And I suspect what we'll do is we'll build the first version without putting Indigenous status into the model.
Paul Walton there, who we've dragged down from Darwin to chair a wonderful CCS Symposium last month involving Indigenous collaborators. You had this experience in Darwin with access to dialysis.
More to continue this discussion. Yeah. So we know that on the basis of race, people are denied surgery, not just by your off the record discussions, but we know, for example, that don't get on the kidney transplant waiting list. We know that there are large differential biases against areas of cardiothoracic surgery and cardiac procedures post cardiac events, not just in the Northern Territory but nationally. So there's good data suggesting there's a bias at play that is not accounted for by comorbidity age or other factors. And of course, there's a broader discussion which has developed by which includes many Indigenous academics that, of course, race is actually a marker. Race is not the issue. Racism is the dominant discourse. And that actually, if we are including race, we're actually including a variable that measures racism. And so now I understand, because obviously I'm a clinician and kidney specialist, that want to tease out some biological factors and indeed socioeconomic factors and remoteness factors, which you talked about. But it's not just for political research reasons, but actually the thought experiments are actually convoluted and complex, and developing a statistical model that teases out race, remoteness, socioeconomic status is not easy, and no one's done it yet. Not only with perioperative issues, but it's a worthwhile discussion. Obviously, somebody's got to give, as Paul pointed out.
Nicola, I think you had a question.
Got one online.
We've got one online, so maybe Nicola first.
Yeah.
Who's on?
Harry,
Sure, yes, sorry, I saw you.
hi. Thank you.
Hi, Sure.
Thank you. That's a really nice talk. Can I ask you, if it takes that long, which I know it does, to get approvals through AIHW for national data sets, how would you plan to dynamically, year on year, get data to keep track of what's happening?
So I'll just repeat the question. If it takes that long to get a data set through AIHW, how would we plan to dynamically year on year update our model? Thank you, and great question. We probably don't need to update the model every year, but certainly we are going to need to update it perhaps every 2 to 5 years. And I think ultimately we would like to see, we're going to need to have an ongoing agreement for regular updates, ideally under the one ethics approval, so that we can keep that as a rolling, ongoing project. And certainly, it's clear now that we need to, to be starting to talk with them about getting the next data set, because by the time I get the data, it is going to be probably nearly 5 years old getting this model made. So we would be looking at needing, you're exactly right, needing to get our request in from another data set. There's also a bit of slowness in the process in that these data originate at each hospital, and each hospital sends the data into their state health department. I'm not yet quite sure how frequently that is done, but I don't think it's done more than once a year. And then all of the data from each state needs to filter into Canberra, and I'm not sure how often that process takes or how long it is either. So I'm not sure that Canberra would get the data any quicker than, for example, 2 years it's collected at the sites. There's potentially a lot more that could be done to speed up that process.
All right, so for time's sake, we might call the discussion and hold at this stage. Nicola, burning question over lunch. And so thank you again to Paul and Jen for a really wonderful CCS. Thank you everyone who did attend in person. Hopefully we will in future seminars have more people in person. I'll use the excuse that people have forgotten where the hub is during COVID, but look on your Google Maps and you'll find it. Or maybe the other excuse is that you're all there hiding in your office waiting for your NHMRC GANT results. And if that's the case, best of luck to everybody. And for those who are here, we do have some lunch outside. Thank you. Oh, next seminar in 2 weeks' time is the Department of Neuroscience. And that is going to be focused on cognitive neurology. One of our star new recruits, Professor Amy Brockman, as well as Associate Professor David Darby will be presenting. So please support that. Thank you.